Hi gang, we've got section 20.2, first hopefully really short video here on gel electrophoresis, and kind of our scenario. If we are dealing with DNA and we have amplified DNA one way or another, either we put it into some sort of a bacteria, into a plasmid, and then let the bacteria be the multiplier, or maybe we did PCR, which we learned about in the last video, or maybe you read about using phages to make large quantities of DNA. Anyway, if we've done that, then we are going to want to study those things at some point. That's the reason that we did this stuff with the DNA a lot of times, is to study the DNA. That's why we amplified it, made lots of copies. And yeah, so we need to have a way of isolating whatever it was we amplified. So maybe it has been inserted into a bacterial genome through a plasmid, into a plasmid. And we need to get it out of the plasmid if we're really going to study it. Or maybe we did PCR technology, and we've got basically a vial full, or a little tube full of all kinds of nucleotides and like partially completed lengths of DNA that we're looking for, and then also some of the DNA that we hoped to amplify. So anyway, one of the ways that we can identify the particular segment that we want and study it in whatever way we want to is through gel electrophoresis, and the idea here is using agarose gel is what they use. It's called agarose gel, and it is somewhat of a fibrous or porous material, and you can use it to separate molecules based on size. You can use it to separate proteins, you can use it to separate DNA, you can use it to separate carbohydrates technically if you wanted to. So a lot of things you can use it to separate. Okay. The philosophy on this one is this isn't going to be exactly accurate, but if I looked at the microscopic level of the agarose gel, okay, it's going to be basically a whole bunch of fibers that are overlapping, okay, and that means that there are spaces in between these fibers, little spaces in between these fibers, okay, of this gel. And if DNA is the molecule that I'm trying to separate, and I find a way to have it pass through this gel with all the spaces that are there, these spaces are going to cause large molecules to slow down as they go through there, because they're going to get hung up on the fibers. Smaller molecules of DNA are going to go through there pretty quickly because they don't get hung up on the fibers as well. And this happens in a very predictable manner, that large molecules of DNA move slowly through the gel, and small ones move quickly. So what they do is they make these gels, and they'll put them into a liquid solution that has some ions in it, some form of an ion solution, and you can load DNA, so a small amount of DNA on one side of the gel here, whatever the DNA is. If we go back to my experiment with the bird malaria, so we had these, after the PCR was done, we had these little tubes, and we hoped that we had made copies of the malaria, but we weren't sure if we had made copies of the malaria genome, or the gene that we were looking for there. And so what we did is we loaded it into the ends of the gel, okay, and this would have been laying flat on a table, okay, so you're looking straight down on it as it's flat in a table, and we load these things in here, and then we run electricity through the liquid that they're in, and that electricity, we put the negative side of the electricity over here, the positive at the opposite end of the gel, and DNA has a negative charge, and so it will be attracted to positive. So as the electricity flowed through the water here, the DNA would be drawn towards the positive electrode, or the cathode. Anyway, large molecules of DNA would move slowly, smaller molecules would move more quickly. You have to come up with a way of staining the DNA when it's all said and done, and we have ways to do that. But we could look at this, and so when I was doing my bird malaria research, what I knew is I was looking for a section of DNA that was long enough that maybe it was this length right here. And so I'd run a gel and look at the picture, and if I saw a band here at this length, okay, then I knew that, okay, this one has a malaria, and I amplified the malaria. This one also has a malaria, and I amplified the malaria. This one does not, because it didn't have that band at the right spot. Okay, and so I could identify that. Then, if I wanted to, and this is what we did, if I further wanted to study specifically the malaria gene that I was looking at, I could cut this out of the gel. The gel is kind of like, it's almost like Jell-O, like a really thick Jell-O. So I could cut it out of the Jell-O, okay, isolate the DNA from the gel, Okay, and then I could do studies on what we did. We sequenced the DNA, so we actually looked at the different sequences of malaria from different places, the genetic sequences, which we'll talk about at a future time here, how to sequence DNA. But anyway, so that's gel electrophoresis. This is only a 5-6 minute video here. I'm actually going to stop it right here. I'm going to do a separate video completely that talks about how you can use gel electrophoresis specifically to identify different types of genes, alleles, and things like that within a gene. And yeah, hopefully this was good for you.